Hello, everyone, and welcome to Lab 13, experiment number 9, the titration of vinegar. So there's lots of background information for this experiment. Let's start with the pH scale. So the term pH stands for the power of hydrogen and is a number between 0 and 14. And it represents the concentration of hydronium, which is also read H3O+, for different solutions. The pH scale can be used to describe the acidity or the basicity of solutions. A neutral solution has a pH of 7.0 at room temperature. An acidic solution has a pH less than 7, and a basic solution has a pH greater than 7. And recall that when we're talking about the concentration of hydronium ion or H3O+, is formed when water reacts with a proton, which is H+. So because we're dealing with an aqueous or water-based solution, we can think of H+ as being the same thing or equivalent to H3O+. So again, pH is a number between 0 and 14. It tells us whether or not a solution is neutral, acidic, or basic. Again, pH is less than 7 are acidic, greater than 7 are basic, and the pH is related to the concentration of hydronium ion in the solution. So at a pH of 7, the concentration of hydronium ion is 1.0 times 10 to the negative 7 molar. In an acidic solution, the concentration of hydronium ion will be greater than that. So it will be, for example, 1.0 times 10 to the negative 5. Remember that that's actually a larger number because it's a smaller exponent. And then a basic solution will have a concentration of hydronium ion less than 1.0 times 10 to the negative 7. The pH of a solution can be measured different ways. It can be measured accurately using a pH meter. We can also use pH paper, which is what we're going to be using in this experiment. It's a little less accurate. You can see we can't get multiple significant figures, right? But we can get a general idea of the pH using pH paper. And we can also use a variety of indicators, which are compounds that turn different colors that correspond to different pH values. There are many common substances that exist at different pHs. Some are acidic, some are neutral, and some are basic. So for example, gastric juice, which would be the liquid in your stomach, is very acidic, right? At a pH of 1.6. Lemon juice, vinegar, soda, and citrus, those are all very acidic substances. And then we have, on the other end of the scale, some very basic substances. For example, bleach, sodium hydroxide solutions such as lye, milk of magnesia, or even Windex, right? Or any other similar window cleaner that contains ammonia. So how do we calculate the pH of solutions? Well, we talked about how the pH was related to the concentration of hydronium ion. So the pH can be calculated as the negative log of the concentration of hydronium ion. So it's a logarithmic scale, base 10, that corresponds to the concentration of hydronium ion in aqueous solutions. The negative powers of 10 in the concentration, for example, if the concentration of hydronium ion was 1.0 times 10 to the negative 2, that -2 is a negative power of 10 in that molar concentration of hydronium ion, the negative log turns that exponent, that -2 exponent, into a positive number. So then that becomes a pH of 2. We write 2.00 because the significant figures is determined based on the concentration of hydronium ion, so the number of significant figures in the coefficient. So for example, 1.0 has 2 significant figures. So if we're taking the negative log of 1.0 times 10 to the -2, and we see what our calculator gives us, even if our calculator only says 2, we need to write 2.00 because 2 significant figures in the 1.0 means that we should have 2 decimal places after the pH value. So the 2 comes from the exponent, and then the additional significant figures come from the significant figures in the concentration of the hydronium ion. And then it's important to note that we can also reverse that. So later we'll talk about, given the pH value, going back to the concentration of hydronium ion. So for example, if you're given pH of 2.00, you need to report your concentration of hydronium ion to only 2 significant figures because of the 2 places after the decimal in the pH value. We'll do some calculation practice later to make sure that we practice that skill. So it's important to recognize that the pH scale is a 10-fold change. So when you go from a pH of 2 to 3, that is a 10-fold change in the concentration of hydronium ion. So the concentration of hydronium ion went from 10 to the -2 to 10 to the -3, which is a 10-fold change. And that's represented by a 1-fold change in the pH unit. So it looks like they're pretty close together, but the concentration is actually 10 times higher with a lower pH value. Concentration meaning the concentration of hydronium ion is 10 times higher as you move one lower in the pH scale. So going from 3 to 2, our concentration of hydronium ion is 10 times higher. So we can also reverse the process like I was talking about before. And if we're given the pH value of a solution, we can calculate the concentration of hydronium ion, or H3O+. We do that using this equation. The concentration of hydronium ion equals 10 to the negative pH. So if we're given the pH, we can just take 10 to the negative of that pH to get the concentration of hydronium ion. I would recommend that you take your calculator, pause the video, make sure that you can input 10 to the negative 3.42 and that you are getting this concentration of hydronium ion equals 3.8 times 10 to the negative 4 in your calculator. Notice that there are 2 places after the decimal in the pH value that's given. We were given 3.42. So because of the 0.42, that's 2 places after the decimal, our concentration of hydronium ion will only have 2 significant figures. So we'll report it as 3.8 times 10 to the negative 4. So how can we relate the concentration of H3O+ or hydronium ion to the concentration of OH- or hydroxide ion? So in a neutral solution, the concentration of hydronium ion and hydroxide ion are equal. But in an acidic solution, there is more hydronium ion, and that means that there's corresponding less amount of hydroxide ion in solution. In a basic solution, there is less hydronium ion and correspondingly more hydroxide ion in solution. So how can we then use an equation to relate the concentration of hydronium ion and the concentration of hydroxide ion? Well, we can use the expression using Kw. Kw is called the water dissociation constant, and it's a constant that is equal to 1.0 times 10 to the negative 14. So that constant is equal to the concentration of hydronium ion times the concentration of hydroxide ion. Because like we saw in the last slide, as the concentration of hydronium ion goes up, the concentration of hydroxide ion goes down proportionally. And the product of those two is always the same constant, and that we call Kw. So we can use these equations, and you can see it's just solved for either Kw, hydroxide ion concentration, or hydronium ion concentration. We can use this to, anytime we know one, either we know the hydronium ion concentration, and then we can use this equation to find the hydroxide ion concentration, or if we know the hydroxide ion concentration, we can use the equation to find the hydronium ion concentration. Here's a table that shows some examples of hydronium ion concentration and hydroxide ion concentration in different neutral, acidic, or basic solutions. So we can see here in a neutral solution, the concentration of hydronium ion and hydroxide ion are equal, and they're both equal to 1.0 times 10 to the negative 7 molar. The product of these two is 1.0 times 10 to the negative 14, which is our constant Kw. The next two examples are acidic solutions. Notice that there is significantly more hydronium ion than there is hydroxide ion in these solutions. We can tell that because of the smaller exponent. Note that 10 to the negative 5 and 10 to the negative 2 are larger than 10 to the negative 7, which is larger than 10 to the negative 10 or 10 to the negative 12. So these would be some acidic solutions with higher hydronium ion concentrations relative to hydroxide ion concentrations. Regardless, though, the product of the hydronium ion concentration and the hydroxide ion concentration, which just means the answer to those two numbers multiplied together, that's the product, is always going to be the constant Kw. In a basic solution, we will have relatively larger amounts of hydroxide ion concentration, or it will be larger than 1.0 times 10 to the negative 7. Note that 10 to the negative 6 and 10 to the negative 4 are larger than 10 to the negative 7. So there's a larger amount of hydroxide ion and a correspondingly smaller amount of hydronium ion in those basic solutions. And again, we can, given one of these values, we can calculate the other using the equation where these two multiplied together is always equal to this constant. Here's another table from your book, and it's a comparison of pH values at room temperature and the hydronium ion concentration and hydroxide ion concentration. Remember that a pH of 7 is neutral. Anything with a pH lower than 7 is an acidic solution. So those are going to have correspondingly higher amounts of hydronium ion and lower amounts of hydroxide ion. Any pH larger than 7 is a basic solution, and those will have correspondingly larger concentrations of hydroxide and smaller concentrations of hydronium. So let's do some practice. What is the pH of an ammonia solution with a hydroxide ion concentration of 3.7 times 10 to the negative 3 molar? So what is it giving us, right? The question is giving us the hydroxide ion concentration. And it's asking us for the pH of the solution. In order to calculate the pH of a solution, we need to know the hydronium ion concentration because pH is calculated as the negative log of the hydronium ion concentration. So we need to know the hydronium ion concentration, but we are given the hydroxide ion concentration. Luckily, we know that given the hydroxide ion concentration, we can calculate the hydronium ion concentration, and from that, we can calculate the pH. So first, we determine the hydronium ion concentration by taking Kw, which is 1.0 times 10 to the negative 14, and dividing it by the hydroxide ion concentration, which is given to us as 3.7 times 10 to the negative 3. This gives us 2.7 times 10 to the negative 12 as our concentration of hydronium ion. Once we have that, we can plug that into the negative log of the hydronium ion concentration equation to calculate pH. Recall that because we have the 2.7 or two significant figures in the coefficient in the hydronium ion concentration, we need to have two places past the decimal for the number of significant figures in the pH value. And the 11 came from the exponent. So we record our answer as 11.57 to the correct number of significant figures for the pH of the solution. Let's do another sample problem. We're going to be calculating hydronium ion concentration from pH. So recall that if we're given the pH, we can calculate the hydronium ion concentration because we know that the concentration of hydronium ion is equal to 10 to the negative pH. So we are given the pH, we want the hydronium ion concentration. The equation that we're going to use is hydronium ion concentration is equal to 10 to the negative pH. So we input into our calculator 10 to the negative 7.5. I think you should practice with your calculator to make sure you're getting the same answer here. And you should be getting 3 times 10 to the negative 8 molar for the hydronium ion concentration. Notice that our pH was given to us with two significant figures or one number past the decimal. Because there's only one number past the decimal, we can only have one significant figure in the coefficient of the hydronium ion concentration. Now, this question was not asking us to calculate the hydroxide ion concentration, but if it was, we could do that using the equation with Kw. All right, let's move on from the pH equations to talk about the difference between a strong and weak acid in terms of their dissociation in water. So if we mix a strong acid in water, for example, HI or HCl, it will completely dissociate into hydronium ion and iodide ion. Now, we are just writing hydronium ion because we mixed it in water. So the H plus from HI is combining with water to form H3O plus. But notice that the level or the concentration implied by this bar graph is that this is entirely converted to the two ions and no HI remains. It's a very different situation with a weak acid. For example, HF is a weak acid, and it does not fully dissociate in an aqueous solution. And in fact, only a small percentage of it dissociates into hydronium ion and fluoride ion. Most of it stays as HF intact because it's a weak acid, so it has a hard time donating its H plus ion. Let's talk about the relative strength of some different acids and bases. So again, some are categorized in the strong acid category and weak base category, and those are related as the acid and conjugate base. And then some are categorized as weak acids, and when they form their conjugate base, it would be a strong base because those two are reciprocally related. So in this experiment, we're going to be using acetic acid, which is a weak acid. And so that's just something to note, because a lot of times when you look at or are talking about a titration, a lot of times they're talking about a strong acid, but we're going to be using a weak acid. But in our case, it's not really going to make a significant difference. Let's talk about the reaction, because a titration is essentially a neutralization reaction. We're going to be treating an acidic solution with a basic solution, and the acid is going to be reacting with the base. And we call this a neutralization reaction because the acid and base react together to form water and an ionic salt. The water comes from the H+ that's given from the acid and the OH- that's given from the base. So H+ and OH- combine together to form water. The counter ions, which are Cl-, so chloride ion, and Na+, sodium cation, those are going to combine to form an ionic salt. The same type of reaction is what we're going to be doing in this experiment, except for instead of using hydrochloric acid, which would be more difficult to work with at home because of the strength of the acid and the danger that involves, we're going to be using acetic acid, which is a weaker acid instead. But acetic acid still donates H+ ion, which can react with OH- from sodium hydroxide to form water. And then instead of sodium chloride, we form sodium acetate. But it's the same sort of neutralization reaction occurring. It's just that we're using a weaker acid. So in an acid-base neutralization reaction, one H+ is going to react with one OH-. So if we write the strong acid and the strong base as ions, we separate the H+ from the Cl-, and we separate the Na+ from the, this should say sodium hydroxide. And then this is an error from your book. This should be NaCl plus sodium hydroxide gets converted to water, and I don't even know what I just said, sorry. HCl, aqueous, plus aqueous sodium hydroxide forms water and aqueous sodium chloride. So this reaction is actually written incorrectly. I apologize for that. I didn't catch that before. That's why there's hydroxide over here in the ionic equation, the total ionic equation, because this should be hydroxide over here, sodium hydroxide, and this should be sodium chloride. So just switch those two in your notes. So if we separate them into ions, we see that the sodium cation and the chloride anion are just spectator ions, and that the net reaction really involves the H+, which we often just call a proton or hydrogen ion or hydrogen cation, is reacting with sodium hydroxide, or just hydroxide if you are just looking at the OH- ion, and it forms water. So that is an overall neutralization, because the positive and the negative charges have gone away into a neutral product, right? And it boils down to, this is basically the net ionic equation. And notice that would be the same regardless if this is HCl or acetic acid. So let's talk about the titration that you're going to be doing in this experiment. So titrations typically involve a known volume of acid that's placed in a flask, right? So this is an Erlenmeyer flask, and you can see it has a clear solution in it. And we would know the volume of the acid solution that we placed in the Erlenmeyer flask. What we don't know is the concentration of the acid in that Erlenmeyer flask. We know it's in acidic solution. We know the volume, but we don't know the concentration of the acid. That's what we're going to be solving for. This is a burette, and inside of it was placed aqueous sodium hydroxide. So a solution of known molarity sodium hydroxide would be placed inside this burette. But in our case, we're not going to be using this fancy equipment. It would be much too expensive to put that in your kit. So instead, we're going to use a Burrell pipette, and we're going to be calibrating the volume of a drop using that pipette. So that's a way to avoid using the more expensive equipment that's shown in this picture here. Okay, so again, we know the concentration of the sodium hydroxide solution in this pipette, and we know the volume of the acid solution we put inside the Erlenmeyer flask. We're going to be dropwise adding the sodium hydroxide solution into the Erlenmeyer flask, and as hydroxide is introduced to the solution, it is reacting with the H+ ions or the H3O+ ions in the Erlenmeyer flask from the acid solution. And it is undergoing that neutralization or acid-base reaction, right, where H+ is reacting with OH- to form water. So eventually, as we add more and more base, there's going to be less and less H+ for it to react with. So once we've added a certain volume of sodium hydroxide, it will have completely reacted with all of the H+ ions in solution. And that, we say, is the equivalence point. However, we don't actually measure the equivalence point, we measure the end point. And the end point is when we see a persistent pink color to the solution in the Erlenmeyer flask, or in your case, it's going to be in a test tube. So as the sodium hydroxide gets introduced, a pink color will appear, but as the sodium hydroxide has a chance to swirl around and react with the H+ ions, the color will go away. The color is from an indicator. We actually put a drop of phenolphthalein indicator inside that Erlenmeyer flask, and it's colorless before the endpoint. So at the solution, when it's still acidic, is colorless. But once it reaches that endpoint and there's excess hydroxide ion in there, a pink color persists. So when we have a light pink color that won't go away, no matter how much swirling we do, that's the end point of the titration. So we can know how much sodium hydroxide was added because this is a calibrated, you can think of it as almost like a graduated cylinder, right? So there's volume measurements here. We can take an initial volume and a final volume, and the difference between those is the volume of sodium hydroxide added. Now, again, you're not going to be using a burette, you're going to be using a Burrell pipette. But you can still count the number of drops that it takes you to get to the endpoint, and you will have done a calibration where you'll know exactly how many drops is equivalent to a milliliter using that Burrell pipette. So we'll still be able to get the volume of the sodium hydroxide solution added to get to the endpoint. So again, a titration is a laboratory procedure that's used to determine the molarity of an acidic solution. We use a base, usually a strong base such as sodium hydroxide, to neutralize the acid in the acidic solution. And we are going to know both the concentration of the sodium hydroxide solution, the amount that we had to add of the sodium hydroxide solution, and we also know the measured volume of the acid solution that we initially put into the Erlenmeyer flask or our test tube. The titration does require a drop of indicator, and a great indicator for this particular titration is phenolphthalein because the color change happens near the equivalence point. So again, the acid solution is in the Erlenmeyer flask in this picture. It's going to be in a test tube in our experiment. And the sodium hydroxide solution is going to be what's in our Burrell pipette, right? And it's also shown in the burette in this picture, right? So in the following titration, a specific volume of the acidic solution, so we would know the exact volume that we put in here, is titrated to the end point. And we say at the end point, the amount of moles of sodium hydroxide that was required or added to neutralize the acid is equal to the number of moles of acid in the test tube or Erlenmeyer flask. And we know the concentration of sodium hydroxide, so we're going to be able to calculate the concentration of the acid solution. Again, we're going to use the indicator phenolphthalein. It is used to identify the endpoint because of the color change that occurs very near the equivalence point. And it turns pink when the solution is neutralized. So at the endpoint of the titration, the moles of base added are roughly equal to the moles of the acid in the solution. The concentration of the base is known. That's provided to you on the bottle of sodium hydroxide. For example, my bottle says 0.4 molar sodium hydroxide. We know the volume of the base used to reach the endpoint. That's going to be, in our case, the number of drops we had to add of the sodium hydroxide to get to the endpoint or the pink color to persist. And from all of that, we're going to be able to calculate the molarity of the acid for the neutralization reaction. So let's look at an example calculation. So it says, what is the molarity of an HCl solution? So that's what we need. We need the concentration or the molarity of a hydrochloric acid solution. So that's the acid solution. If 18.5 milliliters, that's a volume of 0.225 molar sodium hydroxide. So notice that they've given us the volume, 18.5 milliliters, of the sodium hydroxide solution. They've given us the concentration of the sodium hydroxide solution as 0.225 molar. And it says, what is the molarity that is required to neutralize 0.0100 liters of the HCl solution? So that's the volume of the HCl solution that we added to the Erlenmeyer flask or the test tube. So note that we're given lots of information here. We're given the concentration of the sodium hydroxide solution, the volume that we had to add to get to the endpoint, which is 18.5 milliliters of that base solution. We're given the volume of the acid solution. So again, that was 0.01 liters, right? Or 10 milliliters of the HCl solution. And what we need is the molarity of the HCl solution. We need to look and know the balanced chemical equation. We need to know that there's only one H plus in the acid reacting with one OH minus to form water. So the number of moles of OH minus added are going to be equal to the number of moles of H plus that were reacted in this neutralization reaction. So we want to write a plan to calculate what it's asking us to calculate, which is the molarity of the acid solution. So we're going to start with the volume of the sodium hydroxide solution. 18.5 milliliters. Because we're going to eventually want to use the concentration of sodium hydroxide, which is molarity, which is moles over liters, we're going to convert milliliters to liters. So we'll start with the 18.5 milliliters of the sodium hydroxide solution and use the metric conversion factor of 1,000 milliliters is equal to 1 liter to get the number of liters of sodium hydroxide solution used. In order to get the number of moles of sodium hydroxide solution, we need to use the molarity of the sodium hydroxide solution, which is the 0.225 molar, as a conversion factor. So we want to remember that capital M, which stands for molar or molarity, is equal to moles over liters. So moles of sodium hydroxide over liters of solution. So we'll be able to use that as a conversion factor to go from the liters of the sodium hydroxide solution to the moles of sodium hydroxide. We use 1-to-1, the mole-to-mole conversion factor given to us from the balanced chemical equation. So the number of moles of sodium hydroxide is actually just equal to the number of moles of HCl. And then once we have the number of moles of HCl, we can divide by the liters of the HCl solution. And that was given to us, again, already in liters, 0.0100 liters of HCl. And because we have moles of HCl over liters of the HCl solution, we are at the molarity of the HCl solution. So there are multiple conversion factors that we are going to need. Because we have to convert milliliters of the sodium hydroxide solution to liters of sodium hydroxide solution, we need to note that 1 liter is equal to 1,000 milliliters. Remember that we can use concentrations in molarity as a conversion factor because they relate the number of moles of something, for example, sodium hydroxide, in the number of liters of the sodium hydroxide solution. So the 0.225 molar sodium hydroxide is 0.225 moles of sodium hydroxide in 1 liter of the solution. So we can use that as a conversion factor to convert from moles of sodium hydroxide and liters of sodium hydroxide solution. And then we also need to note that 1 mole of sodium hydroxide is equal to 1 mole of hydrochloric acid, right? That is given to us from the balanced chemical equation. So putting that all together, we start with the 18.5 milliliters of the sodium hydroxide solution used. We convert that to liters of sodium hydroxide by dividing by 1,000 milliliters in 1 liter. We use the molarity of the sodium hydroxide solution, or the 0.225 moles of sodium hydroxide in a liter of solution, in order to get to moles of sodium hydroxide. From moles of sodium hydroxide, we want to get to moles of HCl, and that's just 1-to-1. So since there's a certain, however much moles of sodium hydroxide, that will be equal to the number of moles of HCl, which is the 0.00416 moles of HCl. We divide that by the liters of HCl solution, which was given to us in the equation as 0.0100 liters of HCl. And we get the molarity of the HCl solution, which is 0.416 molar HCl. So that's the type of calculation you're going to need to do in this experiment. All right, speaking of the experiment, let's go ahead and talk about the details of the experiment. So it talks about that you may be given an unknown vinegar solution where it's not told to you what the concentration of acetic acid is in the solution. But actually, you're not going to be given an unknown. You're going to be provided with a solution of known concentration. So you were either provided a 5% or a 10% acetic acid solution. So you're going to record that, right, in the description here in the data sheet. So you're either just going to write a 5% acetic acid solution or a 10% acetic acid solution. And you're going to measure the pH of that solution using pH paper. Again, you can just take a drop of that solution, put it right onto the pH paper. Note the color according to the scale to determine an approximate pH of the solution. You're going to get 3 Burrell pipettes out and label them T, A, and W. T will stand for the titrant, and we will use that for the sodium hydroxide solution. A stands for acid, and we'll use that for measuring out the 10% or 5% acetic acid solution. And W stands for water, and we'll use that for the calibration of the Burrell pipette at the beginning of the experiment when we're just measuring out drops of water. Okay, so then here's some information about the first photo to take of your acetic acid solution, right? So whatever vials that you were provided, just take a picture of those and include it. All right, moving on to the next part. In part 3 of the procedure, it says to use the Burrell pipette labeled with the letter T. Because you went ahead and got out 3 Burrell pipettes and labeled one W, I think it makes more sense just to use the W pipette, which is just going to be when we use water. So we're just going to be using water in this part. So just go ahead and make a substitution here and use the pipette labeled W. And then we're going to be calibrating the Burrell pipette using our 5 milliliter graduated cylinder. You're going to place drops into the graduated cylinder up to the 3.00 milliliter mark, right? So we're adding drops of just distilled water into your smallest graduated cylinder until the volume reaches 3.00 milliliters. Remember that you're measuring from eye level at the bottom of the meniscus, and it needs to be exactly at 3.00 or as close as you can get using a certain number of drops. And you're going to do 3 trials of this. So in between, you'll want to completely dry out your graduated cylinder so that it's completely dry for each trial. And you're going to record, again, the number of drops required to reach that 3.00 milliliter mark on the graduated cylinder. And then you're going to calculate the average for all 3 trials. And so the average number of drops to reach that 3.00 milliliter mark. And then you can just add all 3 of those numbers up. So now it's the number of drops to reach not the 9 milliliters total. Divide by 9 and we'll get the number of drops per 1 milliliter. And this is going to be an important number because we're not able to use a normal burette for this experiment. So we need to know how many drops of sodium hydroxide we use, and then how does drops relate to milliliters of that sodium hydroxide solution. We're making an assumption here that the volume in a drop of water is equal to the volume in a drop of the sodium hydroxide solution, but that assumption is reasonable because sodium hydroxide solution is a very low concentration, so it's mostly water. So the volume in 1 drop of water should be the same in a drop of the sodium hydroxide solution. All right, so moving on, we are going to start working on the actual titration of the acetic acid solution that you were given. So again, we are going to be doing 3 trials, right? Which is why you were given 3 vials. So in my kit, I was given 3 vials of 10% acetic acid solution. And I was given a 0.4 molar sodium hydroxide solution to use as my titrant. I was also given a vial of phenolphthalein, which is again the indicator, and I'll be using 1 drop of that in each test tube. Again, it will be clear when the solution still has H+ ions. And then once there's an excess of OH- ions and all the H+ ions have been neutralized, it will turn to a pink color. Okay, so we want to measure accurately the volume of the acid solution. So you're using your 10% acetic acid solution, but we want to accurately measure the volume that we put into each test tube. And we're going to be using the largest size test tube that you have in your kit that you purchased. And in order to do that, we're going to be using, again, our clean and dry 5 milliliter graduated cylinder. And we're going to quickly pour approximately 1.9 milliliters of the acetic acid solution into the graduated cylinder. So just slightly below the 2 milliliter mark. And then using our Burrell pipette that's been labeled A for acid, we're going to add more of that 10% acetic acid solution dropwise, so that the volume is precisely 2.00 milliliters. Once we're at that precise volume of the 10% acetic acid solution in the 5 milliliter graduated cylinder, we're going to pour that liquid from the graduated cylinder into the clean large test tube. Because there's going to be some residual liquid left behind in the graduated cylinder, you can actually use some distilled water and use your Burrell pipette that's labeled W to get the rest of the residual acetic acid solution from the graduated cylinder into your test tube by just adding a little deionized water to the graduated cylinder, sort of washing it around, and then adding it to the test tube. Now, of course, now you're going to have more than 2.00 milliliters in the test tube, but that's okay, because we know we measured 2.00 milliliters of the acetic acid solution, and the rest is just water. Right, so we're going to use that 2.00 as our volume of the acid solution used. The point of the wash is just to make sure that all of the acetic acid gets into the test tube. So once we have those 3 test tubes, we're going to add in 1 drop of the phenolphthalein indicator into each test tube. It's very important that you don't forget to do that, because if you don't do that, you won't see a color change and you won't know when the endpoint has been reached. All right, so in table 3 in the data sheet, we're going to be recording the percent of vinegar. So again, I would record 10% here from what I was given. The volume of the vinegar solution per trial. So I'm going to write 2.00 milliliters if I followed the directions and used that exact amount in each trial. If for some reason you end up using a different volume of this solution, you would record that here as well. But it's recommended that you use 2.00 milliliters for each trial. And then we're also going to record the molarity of the sodium hydroxide solution, which again comes from this bottle here. So I'll record 0.4 molar sodium hydroxide solution. All right, now that I have all of the acetic acid solution in the test tube, it's time for me to titrate. So I'm going to be adding the sodium hydroxide solution using the Burrell pipette labeled T in order to get to the endpoint where the pink color persists. So what I'm going to do is first condition the Burrell pipette labeled T. And I can do that by just taking that pipette, sucking up some of the sodium hydroxide solution, shaking it all around the inside of the Brill pipette, and then pipetting that directly into the waste container. So that just makes it so that it's conditioned, right? Or every little drop, last drop in there is going to be the sodium hydroxide solution. So we're gonna, after we condition it, we're gonna drop as much sodium hydroxide as we can and titrate the first test tube. We're going to be careful to count the drops of sodium hydroxide that we add, right, into the test tube, 1 drop at a time. Because you're going to add quite a number of drops, it says you can actually go ahead and quickly add the first 35 drops, pause to stir the test tube around, and you might notice a pink color initially, but as the base reacts with the acid, that pink color will go away. If there's any drops that get stuck to the side of the test tube, right, then we want to make sure that those get mixed into the solution in the test tube. And so you can always use a little bit of distilled water at any time to wash down any drops from the Brill pipette with the sodium hydroxide in it. If those get stuck, we can always use water to wash those down into the test tube. So try not to get any drops on the side. Try to go directly into the solution without splattering, and that will make it so that you accurately can measure the number of drops required to get to that endpoint. So we want to count the number of drops, record the number of drops of sodium hydroxide it takes to get to a pink color that will not go away. The lighter the pink, the better for reaching the endpoint. And then we want to use our data from table 2, line C, to go from the number of drops, right, of sodium hydroxide to the volume of sodium hydroxide in milliliters. And so we'll have the volume of sodium hydroxide in milliliters, and it's recommended that you repeat this procedure as long as you have enough solution until you get 3 titrations where the volume of sodium hydroxide added agrees to within 0.5 milliliters. And we can measure the pH of the solution at the endpoint using pH paper. So again, you're going to do multiple trials. You'll take a picture of it once you're done with all of your trials, and you'll see that all of these have been brought to the endpoint where that pink color persists. And we want, again, to have 3 titrations, ideally, that agree to within 0.5 milliliters of sodium hydroxide. And at this point, if you have those, then you can clean up and we can continue working on the calculations, the rest of the calculations for the experiment. So again, we're going to be returning all of the chemicals that you used in all of the experiments at the end of the class to Miramar College so that we can properly dispose of them. So any unused chemicals should remain in their original kit, and anything that else should be poured into our waste container, which is our water bottle, and we can just bring those back to Miramar College for proper disposal. Okay, so moving on to calculating the molarity of the acid, and we are in the calculations in table 5. So we're going to average out the volume of sodium hydroxide. So again, we have at least 3 trials where the volume was within 0.5 milliliter from each other. Take all of that data, average the data, so add it up, divide by however many precise trials you did, and then get the average volume of sodium hydroxide that was required to reach the endpoint. Because that's in milliliters and we need it to be in liters, we're going to go ahead and use a conversion factor of 1,000 milliliters is equal to 1 liter to get to liters of sodium hydroxide that was used. And from that volume of sodium hydroxide solution in liters, we can use that volume and the concentration of the sodium hydroxide solution. Mine was 0.4 molar, so that's what I am using here in this equation. So I'm using that molarity as a conversion factor to go from the liters of the sodium hydroxide solution to the number of moles of sodium hydroxide. And that I'm going to record here in the answer, and I labeled it B. Remember that the number of moles of sodium hydroxide is equal to the number of moles of acetic acid. So these will be the same value. And then we can take the number of moles of acetic acid that you calculated here, B, divide it by the volume of the acetic acid solution you use. So if you use 2.00 milliliters, that's equivalent to 0.00200 liters. And take those, take B, divide it by the volume of the acetic acid solution, and we can calculate the molarity of the acetic acid solution. We can also additionally use the formula M1 V1 equals M2 V2, or for this also works when you're using the molarity and the volume of the base. So that's the sodium hydroxide solution, and the acid because the moles are 1 to 1. So we can solve for the molarity of the acid in this equation as well, because we know the molarity of the basic solution, that's 0.4 molar. We know the volume of sodium hydroxide that we used, right? That's where we got here, A. And we know the volume of the acid solution that we measured out initially. That was the 2 milliliters exactly. And so we can use that to calculate the molarity of the acid. So that's another way to get C here. All right, and then our final table, table number 6, is where we are going to calculate the weight over volume percent of the acid solution and also calculate percent error. So we're going to copy over the moles of acetic acid from the previous table. That was B, so we can record that here. We're going to calculate the molar mass of acetic acid. Again, we can use the periodic table and just calculate from given the number of atoms, right? So the mass of carbon times 2, the mass of hydrogen times 4, the mass of oxygen times 2, right? That will give us the molar mass of acetic acid, and we can record that here in grams per mole. We can use the moles of acetic acid and the molar mass of acetic acid to get the number of grams of acetic acid that were in the solution. And if we have the number of grams of the acetic acid and the volume of the acetic acid solution, we can use that to calculate the mass over volume percent of the acetic acid solution. Again, if you were provided a 10% solution, you would hopefully be getting a value close to 10% here. And we can calculate the error because the known value is what was provided on the vial. If it was 10%, use for the known value 0.10 here. So we just divide by 100. And then take this out of percent by dividing by 100 and use that as your experimental value to calculate the percent error. So at this point, we want to make sure, because this is our last experiment, to clean up the chemicals and equipment, right? So read through these instructions on exactly what to do to clean up at the very end. We're going to be returning some supplies. The used chemicals, any unused chemicals, and some of the Miramar College equipment will come back, and we'll give you more information about that through a Canvas announcement. All right, so that's it. I think I gave you enough information, hopefully, to complete the pre-lab, post-lab, and the experiment. As always, let me know, and hopefully You can come to office hours if you have any questions.